Hey, everybody. I'm here today with Yuri Kim, who is a medical professional from MIT. And she is the lead medical professional in the largest clinical study ever done by MIT with a patient population of, you got it, Lyme disease patients. So we are super honored to have you here today, Yuri Kim, because you're helping bridge the gap between researchers like Dr. Mikki Tal, who we've had on the podcast, patients like the many patients that you serve on a daily basis, and other medical professionals, doctors, all kinds of people who need better information and better research and better findings to help people like myself and everybody listening to this podcast. So it's a complete honor to have you on the podcast, Dr. Kim, and thank you for coming on. And if you could say hello to everybody, please.
Hi, everyone, and thank you, Matt, for giving me this opportunity to really have the conversation with you. My name is Yuri. I am the leading clinical study nurse, research nurse for MIT MAESTRO project. So I work with Dr. Mikki Tal, who's an immunologist at MIT, and we've launched this study that is considered as one of the largest studies at MIT in history, and we're specializing Lyme disease as well as the long COVID. So it's been my absolute honor to be part of this whole entire project that is absolutely meaningful to the field. And I just want to begin by saying that with this largest study, we're very, very close to the end of our study this year, completing enrollment of almost 200 participants so far. So we have an enormous sample, enormous data, and we're very, very excited to close our enrollment and be able to really analyze, to speak up for what patients or our participants have suffered so long without so much of explanation or understanding. So I'm very excited to be part of it, and I'm very honored to speak about my work here.
So you are the perfect person that represents the human side of the cutting edge research because we had people on, they talk to us about this research, but oftentimes they don't understand what it's like to interact with the patient population because they're so in their lab, in their research, under a microscope with all this technology. But with MIT, you and Dr. Tal have this unique perspective because you're dealing with patients in the clinical environment, which is excellent. And I have to say, for everybody listening, that you happened to be at New York City for Fashion Week, where Dr. Tal, Rich, and I walked, and you were there, and my family got to meet you. And unfortunately, I don't think we got to meet there. I may have seen you in passing. We didn't even get to say hello. But my entire family, for the past several months, have been telling me we need to get you on the podcast, just because the short conversation you had with them was so powerful. Because again, you brought that human side of Lyme disease and the compassion. They knew where to get you on to bring hope and positivity to this community. So let's start before we get into the weeds about what you're doing with Maestro there. Tell us about your background and how the heck you got into this world of Lyme disease.
Of course, I would love to start by there. I don't know, man. I don't know how I got ended up in this particular field. My background is I became a nurse about back in like 2015, and I actually started my career as a level 1 trauma center ER nurse, where I really learned about life and death in terms of the about life in general. But this is where I really practiced a lot, and then after that, I actually had some extensive background in the pharmaceutical area before I had become a nurse. So I always wanted to be in some type of research field or pharmaceutical research area with that, because I already had that career. So with the ER nursing background, I just decided to switch my career in more as a research nurse. And I started my research nurse career back in Boston University, where I really learned a lot about various disease and rare disease and Alzheimer's disease. And also I did a very extensive research nursing role in the traumatic brain injury groups type of research. So I have some background in the like rare disease, like neurological disorders, and that made me really interested in about the neuroinflammation in general or other neurological neurodegenerative condition. Well, after that, with the pandemic, I actually relocated myself to South Korea because I really wanted to be with my family. And I really learned a lot about management of a very complex clinical trial in general by working in a clinical research organization company. Well, after a few years later, I had my child and I came back. And with the MIT career, I met Dr. Mikki Tal during the interview. And what she told me about her passion in this Lyme disease and also long COVID, and how she looked at this whole phenomenon in the infection-associated chronic illnesses, how she really is motivated to change the field and really truly help out patients, that really just touched my heart. And I knew that I really wanted to work with her. I really wanted to work for this lab and for this community that where the true advocacy was truly spoken from a scientist. So that's where I started. And it's been 2 years since I joined MIT, and I cannot be happier.
So there's a lot there. I'm going to unpack some of that a little bit more and ask you some follow-up questions there. So I love that you talked about the term infection-associated chronic illness, because that's something I talked about with Dr. Tal in her interview and also when we met in person. And that's something that many people in our community have gotten the federal government and the National Academies of Sciences to recognize chronic Lyme in a paper study just last year, at the end of last year, where they say we call it Lyme IACI now, right? That's the term. So Lyme infection-associated chronic illness, and that's all over now the CDC website, and also the National Academies of Sciences published this paper with advocates and researchers that we've had on this podcast that you know personally who contributed to that paper. So I think that's a really noteworthy topic to discuss. But let's go back even further. You said you had experience in pharmaceutical work before becoming a level 1 trauma center ER nurse. What specifically were you doing in the pharmaceutical space when you first started your career?
Well, the pharmaceutical company, I was working as a medical information specialist. So it's one of the major top global pharmaceutical company, and they have like enormous products. Yeah, you name it, from HIV medication to like a dermatological medication to like allergy medication. So they have all kinds of consumer product and as well as prescription product. My job with the nursing background was to answer any kind of medical related question about the product that they were like using, and to refer them to, hey, you should like speak with the physician about this, consult it, or provide them with the relevant medical information that we have and like article or journal or like a published data or our own internal data to really like communicate with the clinicians who have questions about their product and other medications. So my job was to deliver that related medical information about the product that the pharmaceutical company was selling, to really narrow the gap of the knowledge and just to also provide up-to-date, more like a research where we are in terms of where our research is going, that kind of a relevant information. So that was my background in the pharmaceutical company.
You would essentially advise the pharmaceutical companies from a medical standpoint about how the drugs they were proposing or how the drugs they were using would interact or behave with the patient population, which required you to do additional research to provide that guidance to the pharmaceutical companies.
Yes, and also what I want to say is a lot of clinicians will contact me with some of the medication that were not indicated. The indication was quite different. We call it sometimes off-label indication with the long COVID or chronic Lyme medication treatment regimen. Sometimes some medications is off-label and to be used to treat some of the chronic Lyme symptoms possibly. So a lot of questions were asked about these off-label indications, and my job was to kind of perform the literature search. And of course, from a pharmaceutical perspective, it's safe to say we don't recommend to use it, but this is the research that is proven. So my job was to help out to find out the validated, more relevant information for the clinicians too.
So even way back then, when you first started in the space, you were helping people by guiding them about off-label use for certain pharmaceuticals for things like Lyme disease, and that was right from the beginning. So I'm just kind of building up how you became who you are today, because all of those life experiences equipped you and trained you and prepared you to be where you are today at MIT, right? And so it's so cool to see it all tie together. Yeah. So now you're learning more about off-label use, you're guiding about certain pharmaceuticals, and you decide, I want to go get even more hands-on, and I'm going to become a level 1 trauma center ER nurse. That must have been an experience. I mean, I often tell people in my workplace that, look, this isn't a life or death situation. I work in IT. I'm like, nobody's going to die. I work, so we're not doctors, we're not nurses, but you were a nurse at a level 1 trauma center. People could die. It was life or death. So give us a little bit about what that was like for you to pivot from the pharmaceutical world to the ER nurse.
Yeah. Well, I want to just extend my conversation from what I remember working in a pharmaceutical company as a medical information specialist. With that first career in the pharma area, I really learned the voices of the patients, really, because patients are trying to seek for answers for their illnesses, and they try to seek for the guidance and advices from the clinicians. But oftentimes clinicians in the hospital, because I'm a nurse, I've been in a hospital setting, clinicians have a lot of time to spend hours with the patients to like dig out all the information. So patients themselves will try to seek for any kind of a guidance information by contacting even the manufacturer, pharmaceutical company. And there, my job was to listen and then write down all the symptoms and what happens when they took this medication, adverse events and everything to find out information. I really learned how to communicate with those people in the devastation of the chronic illnesses from their career. And that always motivated me to become a healthcare professional with the advocacy, strong, heartfelt advocacy towards patients in general. And I jumped in the ER nursing because I just wanted to learn. I wanted to learn, and I knew that in terms of learning the critical medical practice, the ER was the right place for me to be really trained within a short period of time. I was just so eager to jump in the medical field as a nurse. So that's where I started my career. It was Level 1 ER Center in Worcester, Massachusetts, where you can imagine someone without any extensive nursing practice background or experiences. I am facing a trauma patient. I am facing a stroke patient or cardiac arrest patient, or just an absolute true emergency. At the same time, I also see the entrance of people in their chronic illnesses all the time. They have a crash and they ended up in the ER. I also have experience in the midst of what is practiced as a true emergency, like stroke, for example, or myocardial infarction, MI in the cardiac system. Those are the true emergency that level 1 ER needs to deal with. At the same time, the true voice, the devastated voices that are truly spoken in people in the chronic illnesses, I also often saw them in the ER asking for help. I remember because it was not the true emergency cases that is regarded in terms of, because we have our protocol, how it handles the urgent cases. There's like, red flag, like orange flag or green flag in our system. I regret that I did not have enough time to truly just sit down and listen to those patients, because that's the reality of the ER system. But I'm only realizing this as I'm communicating and interacting a lot with our chronic Lyme participants with complex symptoms and their daily life that is really, really poorly understood. And I always hear from our patients that, well, some of them have told me already they have given up from the current medical system. They've tried everything. They have tried everything and they've almost given up their hope. They say the only hope they have in this unexplainable, undescribable illnesses is the research. And that's why people travel from such a distance, like several hours of traveling, flights, or even some people come even from different countries, truly trying to look for the answer for their illnesses. So I can always relate to this from my previous experience and how that really changed myself, my life, as I'm interacting with the patients in chronic Lyme disease.
Right. Now, when you were in the ER, did you ever experience any, the true emergencies, the level 1 side of the trauma center that were related to a tick-borne disease? Because we do know certain conditions can escalate to cardiac issues, Rocky Mountain spotted fever can escalate, but oftentimes they don't escalate to that level. So I'm curious what your experience was like in the hospital when it comes to Lyme and tick-borne diseases.
Well, the thing is, Matt, if I remember correctly, of course, I'm talking about my very first year of a nursing career in the ER. I was not an experienced person. So I don't know the extensive history of every individual, but I was trained to be jumping in like stroke cases or other like trauma cases with the other head nurses to help out that patient. At that time, what I recall is I don't remember any of those cases when it's tick-borne illnesses associated. Perhaps those people, after all the urgent symptoms are handled and taken care of in the ER or in the hospital wards, perhaps they're more referred to talk to the PCP regarding further testing. And this is what I also told from my patients to participants as well, that they got referred to speak further with the PCP. Sometimes some of the physicians will be willing to order some testings for the tick-borne illnesses, or sometimes people just get dismissed thinking that no way this is related to the tick-borne illnesses. So yeah, I'm just sharing my experience, but a lot of times I do think that it's not the first thing that, that is addressed in the ER kind of setting, but later on it's more like a referral related consultation to see if this kind of an urgent situation may have been related to the tick-borne illnesses.
Right. The PCP meaning a primary care physician, so the person's main doctor. And I remember when I first got sick, my doctor unfortunately retired a few months after I got sick, but he referred to himself as my quarterback because I was having all these weird symptoms, and he was fantastic. But I think that's the right mentality to view towards your primary care physician. They should be your quarterback. They should refer you to specialists because they have more niche knowledge, but also they're compartmentalized, where a neurologist is only focusing on the brain, the rheumatologist is only focusing on, the immune system and inflammatory markers. The cardiologist is focusing on the heart. So I think the PCP's job is to piece it all together and play a detective and kind of be the central nervous system of your medical team. And if you don't have a good PCP, it's not going to be as good of an outcome for you. And I think part of it is when we are so in a bad place, we go to the ER, but the ER's job is to ensure that we're in a stable place and refer us out to the specialists in a routine environment. And I think that's kind of what you're describing, is people are coming into you with severe problems. You'd alleviate those, send them home and refer them out. And I think that's a good message for people listening to this podcast. So the next thing is, after that, you said you became I believe you said you were a research nurse at in Boston with diseases, specifically neurological disorders. Is that correct?
What did you say? What's your...
Yeah, I'm trying to piece your timeline together. After the ER that you were in, so when you were a level 1 trauma center ER nurse, I believe the next thing that you moved on to in your career was becoming a research nurse in Boston with diseases like neurological disorders, right?
Exactly.
Okay.
Yes.
So what was that like? Why'd you leave the ER, and what made you go to the research nurse role and focus on neurological disorders?
It's actually just a very simple, straightforward personal reason. With the ER nursing, I did not have any option or seniority, but to choose a night shift only. And I've done this for a while. And after that, I just realized my body is not situated for the night shift. I was very miserable physically, and it just wiped myself out. And I just wanted to get back the quality of life that I missed so much. But it was an absolutely tremendous opportunity for me to learn and get trained in the medical field. So with that, I just decided, you know what? I had a pharmaceutical experience. I was always interested in pharmaceutical researches, other clinical researches that are going on. So why don't I look for some more stable day job, not a night shift, but day shift job, 9 to 5, in the academia. And this is where I found the Boston University Clinical Research Nursing position. And my job at that time was implementing a lot of very complex global clinical trials. And like a lot of major pharmaceutical companies will work with the physicians at Boston Medical Center, where Boston University is affiliated with. And then we implemented a lot of complex cases, and as well as also internal research, such as we did an NFL football player traumatic brain injury, which we call it what's that called? Oh my goodness, I cannot remember the name. Yeah. So with the football player with the traumatic brain injury, we also researched that part as well. And amyloidosis is one common genetic disease that we try to research with the intervention of a pharmaceutical drug. And also we did a lot of Alzheimer cases as well. So it just can me generally be familiar with very complex neuroinflammation or neurological conditions. And I just wanted to continue with more research in the future. So that's how we got started.
I wonder because we often hear about the overlap between Alzheimer's and Lyme and amyloid plaques and Lyme and brain health and Lyme. When you were in this role, did Lyme ever come up? Was there ever a connection at the time? Because I know it wasn't as common back then about the relationship between these things.
Not at all. Not at all. I only wish that I had that background. I had this kind of extensive background back then, because when you look at these families and families who suffer from of another family member who were diagnosed with Alzheimer's disease, and they have some history about it, or some people don't have any kind of genetic history, family history, but it's kind of a first time happen in that family group. And now I'm thinking, if someone were to come with the early symptoms of a potential dementia or Alzheimer's disease, I think the first thing that we really need to start to look at is the possibility of the other root causes, such as infectious disease, possibly causing these damages in early onset, So I do think that I'm still learning it, but this is how I changed my view of the neuroinflammation or neurological disorders, that could be another separate root causes, Well, my answer to your question at the time, no, I've never had encountered any cases and never even heard about when I was practicing that in Boston University. Yeah.
And then you moved on to South Korea when the pandemic hit. And
Mm-hmm.
that's when you had your child. But I'm curious, before you came back to the US while in South Korea, what was awareness like about Lyme disease in South Korea compared to the United States?
Well, my understanding is, again, I'm not the expert, but Lyme disease is not very common in South Korea, but there are other tick-borne illnesses. That's my understanding is. Um, um, in Korea, we call it tsutsugamushi tick-borne illness, but I think, well, I might be able to make a different pronunciation in the US language, English language, but there's a different types of a tick-borne illnesses. What I do really feel though, as I'm doing this research in the United States, especially in this rigorous institution like MIT, I do think there's a fundamental, profound problem in other countries that may have a tick-borne illness-related chronic illnesses everywhere, especially probably rural or like agricultural area. Because every time I do our study, I'm thinking about my country, South Korea, where the agriculture, like farmers, often do their farming, but they're always constantly exposed to tick-borne tick bites, they're probably not even aware that tick bites could cause these very complex chronic illnesses, So I do think that there's still a very profound lack of understanding, awareness of the seriousness of a tick-borne illnesses in general in South Korea. That's how I look at it, and more research needs to be done.
Yeah, and to your point, I mean, there are so many different tick-borne pathogens. Lyme is just one of them. When we talk about Lyme disease on Tick Boot Camp, we mean like the holistic picture of all tick-borne diseases, because when I was sick with chronic Lyme disease, I had multiple co-infections, but I didn't know it. But we called it Lyme disease. Mostly everybody in this community, they say I have Lyme disease, but they generally have a lot of other things too from the tick bite and other conditions. So, we need to be mindful that, the study just came out again a couple of weeks ago that built upon past studies showing that different strains, I think they call them genospecies of the Lyme bacteria, cause different symptomology in patients. So some genotypes of Borrelia will cause rheumatological symptoms only, whereas other strains or genospecies are more likely to cause neurological implications. So, brain implications. And I think we're still learning about that. So I think, as time goes on, we're going to realize in other countries, South Korea, we've had people from Japan on the podcast, all around the world, the ticks and the pathogens they carry and the genospecies of those pathogens are going to impact people differently. And I think this is going to open our eyes to the bigger picture as we advance research here in the States with what you're doing at MIT.
Yeah, I definitely think so too. This is a very beginning, which is a really exciting for us to really launch this enormous data. I agree with you, Beth, because I'm hearing a lot about symptoms that are very common in some groups of participants whom I met, and then also some certain symptoms that are often told, if people say, I had for example, other like, co-infection like Babesia or Bartonella, they tell me the history of their like co-infection, and then I hear certain symptoms, more like a neurological symptom, more like a psychosis-like symptoms, and I gather this information by doing a lot of extensive surveys. And I think next job, when we close our enrollment, we need to really divide the subtypes of group of symptomology. You're exactly right. And to see what are components of their tick-borne illnesses look like, to divide what causes which symptom. And I think this data can be also used for other global trials. I mean, I'm talking about South Korea because this is where I originally come from, but other countries like you said, Japan or even Thailand, it's just a like chronic problem right there. And I think it's only a matter of time with this type of research and data to really raise awareness of the critical need for this type of research in all around the world.
Yeah, and the proof positive just Sunday. So today's Tuesday. So we're looking at 2 days ago. I was in just because this is going to launch in a couple of weeks. So for context, I was at a book launch in New York City
Mm-hmm.
for a good friend, Nicole O'Donnell, who wrote a book, Resilient Hope,
Mm-hmm.
and it was filled. So this event was filled with hundreds of people from the Lyme community, and many of whom I was just interacting with. And it was so fascinating to speak to everybody because whether it was I have Bartonella, I have Lyme and Babesia, there's different groupings of people. So some people were primarily focused on their GI symptoms. My GI symptoms are unbearable. I can't function and I'm miserable. Other people were saying, hey, the pain, the inflammation, the autoimmune like side of it is crippling. Other people were saying, hey, I'm really cognitively impaired. Or they didn't want to say it, but I was speaking to some people's parents and they were saying, hey, my son was fully with it. He was great. He was able to communicate. He was really good at school. He got sick with the tick-borne disease. Now he can barely speak. His thoughts are slow. He's not doing well in school and we're struggling. So it's more neurological and cognitive impairment and a cognitive slowdown. So you're seeing these groupings of people impacted by the same things. And I think that's just further backing up what you just said. And more research is going to be able to help us identify why we have these groupings of people and how to help them. And that's exactly what you're doing. So let's let that segue into your return to the States, where you interviewed with the great doctor, Mikki Tal, whom we love. And she's telling you about her passion for Lyme disease and the infection associated chronic illness term and how she's going to change the field, which she and you, it's not just her now, it's Yuri and Mikki. Yuri and Mikki are changing this field. So we couldn't be happier for the two of you partnering together. So walk us through what the interview was like with Dr. Tal, because I tell everybody, I don't know if I shared this with you, when we went, I just told everybody this past Sunday, when we were in the city for Fashion Week, I wasn't nervous to meet all the celebrities. I wasn't nervous to walk on the runway. I was nervous to speak to the researchers. I was nervous to talk to Dr. Tal. I was nervous to talk to Monica Embers. And I remember walking up to Monica Embers and Mikki Tal and getting butterflies in my stomach because I was so anxious to speak to them because they're the real celebrities in the Lyme space. It's, everybody's contributing, but they're the people that are going to change the world. And, so now what was the interview like? And then walk us through day one of walking into the Maestro study and what that was like.
Okay, that's a great question. I'm getting excited as I think about that moment when I spoke with Dr. Tal for the first time. Well, I don't know if this can be public in the podcast, but let me share this actually what exactly how I got connected with her. I already applied for a job at MIT as just a primary care nurse, and there's a MIT medical center in the campus. And I just, with my son being so little, I just wanted to think that, oh, maybe I can just slowly get back to my practice by doing the primary care nursing position. So I got accepted and I already signed my offer letter. I actually emailed Dr. Tal, the team, with my appreciation of this type of work in the field because I was very moved even when I checked their website, our lab team's website. And I just expressed my gratitude for this type of research being done and my sincere apologies that I already took the position and I'm no longer able to, continue with the proceeding with further, And then Dr. Dr. Tal contacted me and she and her team member, we have a lab manager, previous lab manager, her name is Paige. She called me and say, would you still like to speak with us, briefly? And I thought, this research is so meaningful from what I can see. Why don't I just still talk to them? Because I would love to just show my gratitude of this type of research. And after having conversation with Dr. Tal, I'm not a person who kind of turn over things after the last minute. If I signed the offer letter, I'm committed.
Yep.
But she changed my mind. She just moved my heart so much, how she felt about this community and how she really wants to change it. And she's definitely desperately asking for me to join the team together as a research nurse to change this, to bring more hopes to our patient community. I just thought this is more meaningful than I could ever imagine in terms of career in my life, and I want to jump in. And I just decide my mind, change my mind, and the next day, MIT Medical Center will never forgive me and apologize still. But I said I cannot do this position, and then within 3 days, Dr. Tal offered me the position with the offer letter. I signed it right away, and we started off right there. That was the best decision I've ever made in my life, Matt.
That is fantastic. And I'm thankful as well. We are all thankful in our community. So obviously you didn't regret it, but walk us through day one. You walk in the door. What is it like? What are you experiencing? And what are your thoughts?
Day one, well, I clearly remember. And as I'm recalling that moment of my day one of a MAESTRO study, I'm getting very emotional because it reminded me of the one particular patient who was very, very sick, extremely sick, bedbound, with all dysautonomia and other severe fatigue problem. So she's just bedbound. She barely was able to come to MIT to participate our 4-hour MAESTRO study visit. Her visit got even more longer due to the fact that she just couldn't carry on test after another test. I wasn't there at that time. She had already finished her visit, and then it was more like a follow-up Zoom call to see how her PM looks like, or other symptoms or other problems she's experiencing after the study visit. So I was still in the training with our wonderful research nurse. Her name is Tatiana, and then she is one of the research manager at the Clinical Translational Research Center at MIT. So two of us were joining the Zoom call with her, and I was shocked. Matt, I was basically shocked because from my BU, Boston University career as a research nurse, I've never had many people who were truly that much bedbound and not able to even speak clearly because of the severe symptoms. I was very shocked to see my participants lying in the bed, and just her cell phone was tilted because she could barely able to even look at it. The lights were all turned off because of her severe visual sensitivity. Everything will trigger her neurological symptoms. At the same time, I remember that Zoom call was to follow up with the participant to see any other complicated symptom has developed. And she kept saying, is there anything that you need from me? Is there anything that I can contribute? I'm about to go through a certain procedure in a few weeks. Would you need the samples from my, the procedure that I'm about to have? So she's so willing to provide everything she could possibly do to help out this community. Again, Matt, that was my day one MAESTRO with my participants, and in-person visits that I know everybody, all the participants whom I met so far have always had the same motivation. Personally, as a research nurse, I ask them the same question at the end of the visit. The question is, what would you like to know from participating in our research? What do you want to get by participating in our research? And the answer is, what moves me and motivates me so much and makes me so humble is that it's not about them. The answer is, I want to stop this. I want to stop this and prevent this devastating lack of understanding, and just in the midst of chaos of their illnesses, and just like there's nobody to even listen to, nobody to even like try to measure what they've been experiencing in terms of symptoms. They want to stop this. They want to stop the social stigma. So it's about their motivation to help others.
Yeah, and this couldn't depict our community any better. This was our experience and our origin story at Tick Boot Camp. Rich was bit by a tick and didn't know what to do, and I was suffering with chronic Lyme, and my doctors didn't know what to do to help me. So we both reached out together to the Lyme community for guidance, and people with really limited energy, very sick people were hopping onto Zoom calls with us to help us protect our health, Rich's health from his tick bite, and to help me protect my health by teaching me things I can do beyond what my doctors were aware of to help me improve my health. And it's something we talk about to this day because people were so sick. They were in dark rooms. They didn't have cognitive clarity, but they wanted to help and they would do anything they can to help us. So I couldn't agree more. This is a very unique community and a very caring and giving community. Now, I want to take a step to zoom out and really explain how things work where you work, meaning you have the Maestro clinical study, the largest clinical study in MIT history, but then you also have the research lab. So they're kind of separate entities. Can you just explain to us the different groupings of how you operate and how they work together to kind of facilitate research through this clinical work you're doing in Maestro?
Yes, I'm happy to explain that. So within the umbrella of Tal Research Group, and it's by Dr. Mikki Tal. So we have this clinical entity, and I am the leading clinical research nurse of Maestro project, and I'm running this with several other research team member, and we kind of operate the clinical operation together. At the same time, under Tal Research Lab, there's a lot of mouse work and other immunological analysis and other experimental work that we're doing. We have a wonderful team members who are doing the mice experiment. And I have to say, all this mice experiment that we're doing and the human clinical research are very relevant and related together. What we try to do under Tal Research Lab is we try to communicate together on a weekly lab or frequent communication, what we've been seeing from our research participants from their surveys or other measurement that we're running in the Maestro study, and that their mouse experiments they're kind of discussing weekly basis, I do see some similar overlaps. And that really excites us because when they do the mouse experiment, when they infect the mice with the Borrelia Lyme disease pathogen, and the symptoms after several weeks later are kind of the symptoms that some of my patients have reported to me when they recall their first Lyme encounters. So I do think there's a very close relationship and the very exciting work together when we combine these two different entities that we operate separately in the lab. I don't do any type of a mouse experiment here, but I actually think it's going to be amazing, Matt, to also introduce one of our lab members who managed the whole mouse experiment project here too.
Right. And we're not going to go too much into what Dr. Tal does with her mice model testing in the research lab because we've had her on. We actually had two podcast episodes of Dr. Tal. One that was the live in-person interview in New York City. That was a quick 20-minute interview. And then we did a long form one as well. That was about an hour and a half that people can go listen to on our podcast. But really what she's doing there is making observations, testing out some theories, and identifying things to help the larger population, the human population, by testing out theories on mice and then sharing that with you that you can then bring into the clinical world with humans. And what you're learning, you share with her. What she learns, she shares with you. And together, you're constantly sharing information between your two groups. And you're allowing each other to grow at a quicker rate because you're sharing information between the two channels. That really is, again, unique with you because nobody has what you have at another university or research center. So that's what's so special about MIT and the Tal Research Center. So let's now pivot back to you and Maestro, because I think people listening are probably thinking, hey, I'm really sick with Lyme disease. Can I participate? What is it like? What do you do? How do you help people? What are you learning? What are they learning? So, let's talk about, are you enrolling new patients to participate? I guess number one, let's start with that question.
Okay, sure. Well, I introduced myself earlier by saying that I'm very excited that we're very close to the end of our study. So it's been a very exciting couple to almost 3 years since we launched the Maestro study. And our goal is to enroll about 200 participants so far. And I believe we're at about 190 participants at this point, and we are just very, very close. Right now, we are actually going to close out enrollment by filling out some of the participants in their 20s or 50s in long COVID group, So in terms of chronic Lyme cohort, we are absolutely full, and we're very appreciative. So many people have expressed their interest in participation in the study. I do still get a lot of emails from participants who are really willing to travel and really wants to find out more information about their illnesses by participating in our research. I do hope that we are going to launch another project and start to enroll new number of participants. And at that time, we're going to consider all the people who have already applied for MAESTRO study, perhaps put them on the waiting list. So currently, we're very close to the end. And yeah, that's where we are in terms of enrollment.
Is this going to wrap up in the 26 calendar year? Is this like going to end at December 31st of this year kind of thing?
So the goal was actually we were hoping to end our study end of last year, by December last year. But with the holiday season and there was some flu season, I mean, we're still in the high flu season right now. It's been a little bit pushed back, but I am absolutely motivated to close out enrollment by end of February, if not March, to really be able to start processing all the samples that have been aliquoted and securely stored in our MIT freezer. Some of the samples in the research do have some strong batch effect, meaning that we really prefer to ship out all the biological samples and be able to have them run on the same time, by same personnel to just minimize batch effect. So that's why the biological samples have been kind of stored for this long period of time to be able to ship out all together. A lot of our participants who participated previously we're talking about a couple of years ago, they're still very anxious to hear about more publication and results from our study. So I'm as anxious as they are to really close it and be able to provide back the vital critical information that we find here. I'm very excited actually to close it and be able to start analysis.
Right. So when you close it, you're saying you're going to close enrollment and then start analysis. Yeah. So I guess let's ask a follow-up question to that. What is the typical experience like for somebody who's been through your program? So you approve somebody to be a participant in your research study. Mm-hmm. What does that look like from beginning to end as far as their involvement? And even once they're done with participating, what are you doing with their samples that you've collected and how's that going to work in the long term?
Okay, that's a great question, and I'm very excited to even explain that further. Let's imagine that someone's interested in our study, so they're going to visit our website, Tal Research Group website, MAESTRO study site, to fill out the screening survey to begin with, and then our team will sort out the screening surveys to find out eligible participants, and then we reach them out, certain participants, and ask if they're still interested, and then we are going to schedule a consenting call, because with the research, obtaining consent is the number one step to really proceed with anything further. So it's typically done by myself, as a research nurse. I meet with our clinical cohort participants and really just have 1-hour Zoom call to explain what they're going to sign up, what they're going to be doing when they visit MIT or be part of this study. So I give a very extensive information so that they feel comfortable about what they're signing up and answer any kind of questions they may have. At the same time, that moment, day one Zoom call really initiates the close rapport with my participants, because I always love to ask participants, would you mind briefly explaining your journey, because I would love to know individual cases. And this is where I learn. There got to be a reason why 200 participants are giving me a very similar story here. There got to be a common ground here. We need to find this out. So this is where I got really motivated. So after consenting call, what happens is there are going to be multiple survey links to be sent out to the participants. And you know what? Our MAESTRO study surveys are very long. And maybe someday when you visit MIT, I'll also show you what we ask. It's a very extensive questions with a lot of comment boxes. I do add a lot of comment boxes for people to free type everything they need to, because this is a complex phenomenon. This is a very complex illness. It cannot be just answered by one like A, B, C, D, E kind of multiple choice question of survey. It needs patients' experience, what they have gone through in the diary or chronological order, if they can. So I always ask my participant, if you don't mind, would you please share all the time points when this certain symptoms started? If you were to develop GI symptom, when was it? If you were to develop more like dermatological or the MCAS-like symptoms, what was the time point? Because I do think that illness duration really matters a lot. Within the illness duration, we have to track a very detailed, specific time point when certain symptoms started so that we can later on associate this with the biological sample processing and what we find in their like proteomics or other like related data. So that's where what we're trying to do. My job as a research nurse, and this is what I emphasize in our clinical team too, to really find out as much information as a participant. You do have to listen and you do have to take time to really find more information from each participant. So that's what we do throughout the survey. And then after survey, I carefully review that. And then we schedule 1-day study visit at MIT. And this is what I talk about, a 4-hour visit. It's a long day.
And they come on site. So they physically come to you, they travel to you for 4 hours to be assessed by you as a medical professional in an in-person capacity.
Yes, yes. So we do a multiple different test modality throughout the visit, which I would love to even talk more about throughout this podcast. One common symptom that I always hear from a lot of our participants is brain fog, and this is one thing that I'm very passionate about. It comes often with the fatigue, and some people may not even have a fatigue-like symptoms, but they have a severe cognitive dysfunction that they have experienced. So we do test a lot about different symptoms, and what I've learned about from our participant is that brain fog is very complex. It's you have to really ask participants how individual will describe brain fog. We often think that this is a short-term memory issue or concentration issue or other like the word recalling problem. Yes, they're very common. They're very, very common description in terms of brain fog. However, individual will also explain this a little bit differently. Some people say with the light sensitivity or visual stimulus, they have a more brain fog and they experience more brain fog than ever. Some people may say pain causes the brain fog, or some people may say they will explain brain fog as this association of a body and the brain. So this is it seems like a very vague description. Like some like clinicians might wonder like, oh, I don't know how to really take that type of symptom description. But what I really feel passionate about is it's a complex phenomena. Brain fog is not just a memory recall issue, and we really need to find out how individuals describe it and be able to also divide the groups of different description and how we can analyze further. So that's one thing that I really want to work further. The work is still being done, but in MAESTRO study, the way we measure brain fog is we have 3 major testing that we are applying. One is the battery testing that we do. A person can do this remotely a day before study visit, and then we can just really measure the executive function, the type of baseline measurement. And then when they visit MIT, we do 2 major testing. One is eye tracking testing, and the other one is EEG testing. What I want to say to my participants always, and this is somewhat easier to understand, eye tracking testing, we're adding some visual stimulus to see how brain process those visual stimulus and be able to react physically. Versus EEG testing, during EEG testing, we also administer another test called P300. It's a audible testing. So there will be a different sound tone that patients will hear throughout the headset. And then we are able to, we ask those participants to distinguish between the low pitch sound, which we call a common sound, and then the high pitch sound, which is a rare tone sound. So high pitch sound is considered as a stimulus. And we ask participants to tap the space bar every time they hear this high-pitched sound. So while we're trying to measure the brain fog with eye tracking testing in the first place, the next testing is to see how auditory stimulus processing looks like. So it's been very interesting to apply those 2 different testing modalities. And what's striking is it shows a similar patterns in terms of a physical reaction time, and that correlates with the participants' subjective report about their cognitive dysfunction.
So a couple follow-up questions on that one. So the eye tracking, you said that you add stimulus and it shows, there's a lot of consistency between it. So just walk us through exactly what that looks like. So the patient, what is the patient experience? The patient walks into a room, you sit them down. What are they looking at? What's the stimulus? And what are you actually measuring? Give us a little bit more detail on that.
Yes, yes. So the commercial name of the eye tracking device that we apply in our study protocol is called Right Eye. Not that we're using the right eye only, but it's a company name and it looks like a video game like setting. So it's a little computer, and then you'll be relatively in a dark setting room, and you'll ask to sit in front of a computer, and we'll apply the test by pushing the start button. Your job is to follow that written instruction that already kind of measure how you're able to understand what's written and what's asked being the screen, looking at the screen. And then it's going to just measure your basic ocular motor function, such as pursuit, which meaning that the dot will be kind of circling around the screen, and it's going to give you the statement saying, please follow the dot in a circular motion. But please follow the dot in the up and down motion or the other way. And there's also saccade testing, which also evaluates ocular motor function, look back and forth rapidly or up and down rapidly. So those are the basic function testing, but we see that in our clinical cohort, and this is absolutely correlated with their visual symptoms too, the light sensitivity or doubling vision or just visual disturbances in general after their infectious disease chronic illnesses. And they're not able to do this well, especially saccades. I find it interesting that people find it very challenging to look back and forth, even though that particular testing is very basic and just asking your simple ocular motor function. So that's what we're trying to do. And within that testing, it gets continued over and over. And one of the testing is called choice reaction time, where you're going to see different objects. So it's a very straightforward question. The screen will say, do you see I'm just gonna give a very short example. Do you see star shape? Then press 1. Do you see umbrella shape? Then press 2. Do you see flower? Then press 3. And then the screen will keep rotating those images, star and then flower. Randomly, there will be another star on the umbrella. And you're supposed to remember that instruction and be able to produce that physical reaction time. So it's a cognitive function and how one can really perceive the stimulus, be able to make a proper physical reaction time. Compared to our control group, our preliminary analysis show that there's a significant delay in terms of how people are able to process that and be able to produce that physical reaction time. So which correlated their subjective symptom complaints of the cognitive dysfunction. So we're excited that there are ways to measure and capture those symptoms when people are often coming to us by saying they've tried everything, but they're told by their clinicians that it could be possibly related to their depression or anxiety issues, when it could be the problem related to their infection-associated chronic illnesses.
Right. Right. So talk to us about the other one you mentioned, the EEG testing. I think what you said uses the P300 test. And it sounds like that's more of a sound-based test versus an eye-based test. So the eye-based test is you're seeing something on a computer screen, and you explain that brilliantly. But now the EEG testing or the P300 testing is sound-based. Can you give us a little bit more the same way you just did for the eye testing? The patient walks into a room, and what happens?
A patient walk into the room, and the EEG testing that we do in our study, that takes actually the longest time of the 4-hour visit window, takes about 40 minutes. So that single test itself can cause a lot of fatigue for our clinical participants. What we do is we have participants sit comfortably in a chair, and then we're going to use the helmet, and that underneath the helmet, there is a 19 different region that you can measure the brain activity throughout the testing session. And then you're also going to get to wear the headset where those 2 different sound tones will be applied. And this testing has been known many, many decades as a part of the tool. And what we find it interesting is that so what I want to say is that testing, particular P300 testing session is 4 minutes. So 4 minutes, if you think about it, it's not that long, But at the same time, people who complain brain fog or cognitive dysfunction, they find this extremely tiring. And towards the end, the reaction time may be slower and slower, very hard to focus. When you hear the common sound, and I just want to demonstrate an example, if I can use my voice, it would be basically like beep, 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 beep. It's a common sound. Your brain's supposed to ignore it because it's not a stimulus. When I instruct them to distinguish high-pitched sound, out of the common sound, you will hear beep, very high pitch sound. So your brain should wake up and be able to really capture that stimulus and be able to produce the physical reaction time, because I asked them to tap the space bar as soon as they hear the high pitch sound. My participants with the cognitive dysfunction has explained this type of testing very tiring, but also it's like they know, they hear this and their brain, they know in their brain that they're supposed to react right away. But for some reason, to carry into the motor function by tapping the space bar becomes harder. It's like like they're constantly sending their brain signal like, hey, I need to tap the space bar, but it's not as fast as they want to be. So that would be another interesting analysis to look at it together. What I'm very interested in doing this after study enrollment is closed is that combining right eye or comparing right eye, the visual eye tracking testing with the EEG. Would that show the consistent slower reaction time, or would one person do better than the other? If so, actually, let me go back to the survey and how they explain their deep brain fog. Actually, do they have a visual disturbances and those histories, and then they'll do more poorly in the eye tracking test data and do better in the EEG testing, or the other, So I think there's a very interesting analysis that we can look at it together.
Oh, this is fascinating. Okay, so the final thing you talked about, well, really the first, but I kind of went backwards, so I went all over the place with the order. So we talked about the eye tracking, the EEG testing. Now let's talk about the battery testing, because that's done before they come to you physically on site. And I think you said they do it a day before virtually, and it measures executive function. So what's that like? The patient is sitting at home, they open up their computer or their phone, and what are they doing?
Oh, so it's a very straightforward testing, and in fact, it's been a straightforward testing at the same time gives us a very interesting, meaningful value that we're trying to adopt this battery testing, remote battery testing, even to our next research project that we're building on right now, to be done at home. So we're going to launch a study that participants, without having to come to MIT on site, because if you can imagine the 4-hour visit and traveling from long distance, I mean, that itself will cause the PM, So how can we truly reach out to people who are bedbound? Again, as I'm talking about this, it reminds me of the day one, my first participant whom I met via Zoom. How can I reach them out, and how can we get the data from them? Because those are the most critical data that we should really try to combine to find this answer in this chronic illnesses. Because the problem that I always hear our participant is, as much they want to participate, their physical capability becomes an issue, and I want to stop it, or I want to help it out. So that's what I'm trying to say is, this kind of a remote battery testing is very valuable to be also administered when we try to launch the study at home base too. So
And you can scale it. So you can scale it to many more people because it's all done remotely. So I'm with you.
exactly. Especially, I think it adds a tremendous value throughout their basic kind of daily life of all our participants. Imagine that our day will be all different. Some people may have a crash or flare up in certain days. For female, for example, depending where we are in terms of our menstruation cycle or hormone level, we might have a flare up in certain days. We might have a good days. What does that battery testing look like? What the difference looks like? I want to even combine this eye tracking testing to be done at home base. This is what Mikki and I are trying to constantly talking together to see how we can make this possible. Because without having them to come to MIT for a one-day visit, how can we capture everything when they just live their basic, just daily life function, depending on where they are, good days, bad days, what does it look like? What are the difference look like? So that's
Yeah.
where we really
Yeah,
want
because you're capturing them essentially for one day, which could be a bad day, a good day,
to.
probably a bad day because they had to travel to you, and travel can be triggering. So they're coming to you at their most weakest point. So it's
Exactly.
not like a regular blue sky day for them. It's a stressful day for them. So yeah, that makes sense.
I mean, man, I mean, our research center opens at 8 o'clock, from 8 o'clock to 5 o'clock every day. And I do have to ask my participant to arrive by 10 o'clock in the morning at the latest. Usually I ask them to come 9 o'clock in the morning. You can imagine living from distance or like having to get themselves ready to be able to make it down early morning, because I know that certain participants have very difficult time in early morning setting. I think that becomes such a variable and a huge effect on test our data in the research. And I'm learning as I'm running this, and I really want to find ways we can help out participants, but also really have a true, like even better data in the future.
Yeah. Yeah. So just to wrap it up, give us a walkthrough, if you don't mind, what that's like, the battery testing from home. So I open up my computer, I open up my phone. What is it, and what's the patient experience like for that remote test that you do for brain fog?
Very simple. You just sit there, and I'm going to assign a specific code to every individual. And you just need to enter your date of birth, de-identify format, and then you enter the code. And you're going to click by using the mouse on your personal computer. And it's going to ask you a different word memory recalling kind of a testing. It's also going to ask you a like different color. And if the color matches the wording, those are executive functions where you have to make a rapid decision. And then at the end, I believe there will be some number lining testing, trail making testing. So it's from number 1 to 12, for example, or the next testing will be number 1, and you have to relate to A, 2, and then B. You have to like find these like dots together. Again, I find it also really interesting, even that very straightforward, simple trail making testing, people find them exhausting. And really, what's been so fascinating is they always tell me, I know what I'm supposed to click. I know it's there. I know it's there. And I see that, but it's just that the clicking is not as fast as they want it to be. So I do think that the cognitive function and physical reaction time is very kind of closely associated, that we can really compare these outcomes from the battery testing and eye tracking and then the EEG testing altogether.
So, in addition to the brain fog testing, the battery testing, the eye tracking, and the EEG testing, do you do any additional work while they're on site for that 4 hour visit?
Absolutely, yes. So carrying on, we do multiple biological sample, but I'll talk about that later towards the end, because now I'm kind of recalling the sequence of the visit that we typically do. And after the EEG testing, then I have a very special time with our participants by running this capillaroscopy, which is a new devices and new implementation that we've added to the protocol since early last year. So capillaroscopy is a microscope that we look into your nail fold in your finger cuticle area, and we actually even do this in a big toe area. So we look at the capillaries by using this microscope. Capillaries at the very end of your vascular structure, vessels basically. And then what we do is when we look at the capillaries, we try to count the patterns of the clinical cohort, like long COVID chronic Lyme, versus the control group. Everybody, we all have some abnormal shapes or abnormalities in our capillaries, like gigantic capillaries or enlarged capillaries. So those are the terms that it's commonly known. We all have a little bit of that. However, with the chronic illnesses and their vascular structure issues, can we capture something from capillaroscopy? And the answer is, we found this very interesting to see the clinical cohort, to see more abnormal shapes of capillaries. And I'm carefully saying this because this is based on what I've observed by running these capillaries. And the next thing is we're going to run it through the AI analysis and other collaborating with the rheumatologists potentially together. But capillaroscope is something that our patients, participants find it very, very meaningful, because we actually get to look at this together. And what's been very touching when I run this testing with the participants is that participants say that they feel as though they're spoken with all their symptoms they've experienced by looking at this together. And the analysis is upcoming, but I just think that that's a new area that we're trying to understand and also
Huh.
see. Yeah.
So I'm just as I'm listening to you, I'm Googling capillary
Yes.
because I'm trying to visualize what it is. So is it really the skin where the bottom of the fingernail attaches to the finger? Is it the red part in the skin, or is it the skin under the nail? What are you actually looking at?
A capillary is a vessel, the end of our vascular structures, vascular system. So if you can imagine, the cuticle is a very thin area, and this is where the capillaries is located. The end of the capillaries are usually visualized using the microscope. So we're just looking at these very fine branching blood vessels that that form a network between the venous or arteries at the end, and we just kind of see the overall structure and if there's any abnormal shapes that we could identify there. Have been some cases where we see some hemorrhages where we shouldn't really see much hemorrhage in the capillaries, but I've seen a lot of chronic Lyme participants and long COVID participants showing more of these hemorrhages. So it's been a very validating and just very exciting experiment that we've been doing in our protocol so far.
What brought you to even look at that? So what put it on your radar that the capillaries of Lyme patients would be different than the capillaries of it's called the regular population, right?
Yeah, so I always want to refer my PI, Dr. Mikki Tal, who's always motivated with this statement. She always emphasized you have to measure absolutely everything in this particular community because the symptoms are so complex and you cannot miss anything here. We have to look at everything from head to toe. And with the, so much a complaint of the vascular changes that our chronic Lyme participants have experienced, and when you think about even neuroinflammation or neurological disorder, we start to think that is there any way perhaps the neurological symptoms can be associated with the vascular structural problem as well? And if so, how can we identify it? Or how can we capture and identify these things without being so invasive? Or, how can we approach that? And capillary scope was one area that we've always interested in and kind of talked about, and we just decided to implement it.
That's so cool. That's really cool. And I think these can be used to measure somebody's recovery as well. Somebody suffering from chronic Lyme, all these testing, the brain funk test, the battery testing, the eye tracking, the EEG testing, the capillary scopy testing, because they can all be used to show changes. And as somebody's reporting they're feeling better, you could also use these measurable tests to say, yep, the tests are proving you're feeling better, even though you're telling me you're feeling better. That's the way to measure progress, right?
Yes. I want to add one more thing, Matt. In terms of capillary, we are actually doing this in such an interesting way that we usually run NASA lean testing, which is a POTS and dysautonomia testing that we run in our protocol. And NASA lean testing, participant is supposed to be in the supine position, like just lying down flat for 10 minutes. And after 10 minutes, I ask my participant to stand up, lean against the wall. So body is tilted, blood is being pulled, and we're going to measure the vital sign, blood pressure, oxygen level, and heart rate, and really assess very carefully symptoms and signs for the next 10 minutes while participant is standing up tilted. If you could imagine, it's a quite extensive and very exhausting. Person can be absolutely symptomatic. Some people cannot even make it after 3 minutes to 4 minutes, become nauseous and heart rate it's accelerated. It's a task that really needs a lot of attention. We are doing this capillary before the nasaline testing to see what it looks like from baseline. And after that autonomic dysautonomia testing, if the person becomes symptomatic or not symptomatic, we want to compare what it looks like at the end. Does it make any difference, And if it does make significant difference, what is it? And how is it relate to other symptoms? So that's a very interesting way to look in our study by implementing that way. I can't wait to analyze all the data. At the same time, Matt, I want to also say, during this dysautonomia testing, I mentioned that we're measuring vital sign and heart rate and oxygen level. We're also adding a earpiece to be placed in here. The company is called Lumia, where we try to measure proxy cerebral blood flow during dysautonomia testing. And what's been so striking is
Is
that
that, is that like brain blood flow? Is that what that means?
yeah, how much
Okay.
blood flow fluctuates in the brain. So during the dysautonomia testing, if want to become asymptomatic, I would imagine that we'll see accelerated heart rate or like a fluctuated vital signs in general. There have been some cases where I don't see any changes in the heart rate, but participants say, I feel so dizzy, I'm very lightheaded, but heart rate is stable. Blood pressure doesn't show much fluctuation. But I do see some cases where I see a very intensive fluctuation in the cerebral blood flow measurement.
Hmm.
So this could be another added tool in a clinical setting for clinicians to implement when person comes and complains of symptoms of dysautonomia. If the vital sign doesn't show much of changes, perhaps can it be looked at in terms of how much blood flow to the brain changes? I
And
think
that's fascinating. And what's happening there? Is there an increased amount of blood flowing in the brain causing the symptom? Is that what you're seeing in that example you described?
it could be decreased amount of blood flow to the brain that causes this lightheadedness or dizziness too. It should be stable throughout the time. I mean, when you make the posture change from lying down flat to be tilted, of course we're expecting some changes to the blood flow to the brain, but it has to be kind of stabilized after a few minutes. I do see cases where it's like more significantly dropping. Although the vital sign doesn't show any changes. Again, how do we describe that?
Hmm.
Because my participants say feel very lightheaded, absolutely fatigued. But if clinicians were to measure the regular vital sign, it doesn't show anything. So I would imagine in that case, the person would be referred to probably another department or maybe like discuss further consult other department because this doesn't show any other changes. So I do think what we try to really believe in our research is there got to be other ways to capture and measure and make a reliable data for all these chronic and very complex symptoms that they complain.
This is fascinating. So it sounds like you're doing this with all of your individual participants, but once you wrap up the study, I'm ahead of myself. We're gonna come back to the full 4 hours. But once you wrap up the study, you're gonna then do comparisons and try to identify patterns and commonalities to better inform the community. That's kind of what you're doing. You're now building each individual data set for each individual patient, but once you're done, you're gonna have to compare and contrast, right?
Exactly.
Okay, so
Combining
cool.
with their history and every survey responses, I want to just like make interesting case reports of this is what's commonly seen in certain group of people and certain group of clinical cohort. And next thing is we really want to compare everything we capture in our in-person study visit to what we find in the molecular profiling of all the biological samples.
Interesting. We haven't talked about the biological samples yet. We're going to get to that. I know you said that earlier. So let's talk about now. So you talked about the capillary scopy, you talked about the nasaline testing. So capillary scopy is the blood vessels and the fingertips.
Mhm.
Your nasaline testing is your POTS testing. You lay them down for 10 minutes, you sit them up and you make them lean for 10 minutes. And some people can hit, I get it. That's very difficult for Lyme patients. But the fascinating part is sometimes their vitals, their heart, their blood is fine. They're experiencing symptoms, but now with this little earpiece you're putting in can track abnormalities in blood flow in the brain, which is a nice little, I'll call it a tip or something for you to investigate further as you get into this. So what else are you doing in this 4 hour window? Because this is a lot. This is a lot of good stuff. So you're packing in a ton of stuff to this 4 hour window.
This is a lot. And I actually also meet with every participant and spend about 40 minutes to assess their regular vital sign as a baseline level. And also I do apply some neurological assessment that clinicians will do if want to come to the hospital with some neurological symptoms. So we make sure that everything is okay. We just make, or even like as simple as, can you make a facial expression? Can you stick out your tongue and just move side to side? That type of assessment. I want to also emphasize that we're also running the mini mental state exam, which is commonly used to like evaluate Alzheimer's disease, for example. So we're talking about what date is it today, or could you do like what's the latest news you can recall, or can you draw the shape, or can you draw the clock, So those are the common assessment that clinicians will sometimes run in the hospital with our participants who complain this brain fog or neurological symptom. do absolutely fine with that testing. So
Hmm.
what that has taught me as a clinical research nurse is that um, brain fog, all those neurological symptoms associated with the infectious disease can be quite subtle and more complex that those kind of traditional mini mental state exam would not capture anything at all. So there again is a gap between this particular symptom in our clinical cohort and how this can be measured and captured in a clinical setting. So this is where I find it very rewarding that we can really speak that out and voice that out and show the validate data by analyzing all this research testing that we are running in for the 4-hour visit day.
Oh, that is really interesting. Wow.
Yeah.
So what else? What else are we doing in this 4 window? Is there anything else I missed, or is there anything else that we haven't covered yet?
I don't know. I'm getting excited, Matt. Actually, I might have missed something, and if I do, I will come back to you. Maybe even after this podcast done. But
Okay.
if
But you did I want to geek out before we leave this 4 hour window because you
yeah,
did say that we'd get to the biological sample processing. So while they're on site for the 4 hours, you're collecting samples.
mhm.
So can you define what a biological sample is? And it sounds like there's probably many biological samples and what that's like for the patient and what they need to do.
Yes. So biological sample. I just have to circle back our Dr. Mikki Tal's motivation. You have to measure everything. So we do biological collection, sample collection from head to toe. Let's start with the oral sample. So we do a couple of oral sample to just, it's a basically very thick cotton ball that we ask participant to chew for 2 minutes. And there's like a little toothbrush like material that I ask participant to kind of rub their gum back and forth inside of the mouth for a couple of minutes. So we try to just test out antibodies and also try to test the cortisol level. And there are other things that Dr. Mikki Tal is planning in terms of us testing those biological sample, which I don't have much information at this time, but that's what we're doing. And we also do a multiple tubes of a blood collection to see the different antibody testing we run. And also we do the vaginal sample for female participants and rectal samples for both female and male participants. We are in the biological engineering department at MIT, and we're in the center of a gynecological research department. So we are also very interested in to see the effects of the impacts of the infectious disease in a gynecological condition in female particularly. So we really also want to learn about the sex differences and how the response to the infectious diseases, why it's different between men and female. I mean, male and female. So we're going to compare all these sex differences and gynecological issues by collecting all these blood samples, the vaginal and rectal samples from our participants, and to also even look at the gut microbiome possibly. So there's extensive work that we're doing here. Once those samples are collected, what we do is we carry these samples back to our lab and where our web lab bench is, and then we're going to hand out to our laboratory team members who will be carefully allocate all these precious samples, and then we then securely freeze them in the minus 80-degree freezer usually, because that's where the sample can be just preserved intact for a long period of time. And we are going to send out these samples to multiple collaborators that we're working with, academia and all the institution or companies. We're going to look into multiple aspects of those samples. We're looking at proteomics. We are also looking at the so basically we're gonna look at all the proteins possibly and cytokines, how immune function is dysregulated for certain participants.
So proteomics is looking for cytokines in the blood. Can you give us a little bit more on that?
That's a part of it. And the proteomics, we're just basically looking at all the proteins in our body, and we use the Olink proteomic data right now, and the biological samples. So protein is examined throughout that sample profiling, and then that's cytokine. I believe that's part of the proteomic sample. I am not an immunologist, so I don't know the whole extensive kind of list of what we are trying to look at in detail here. But that's something that we can really address by talking with the Dr. Mikki Tal. We're going
Right,
to
but that's why you guys complement each other because you have different purposes, different roles, different expertises, and together you guys are going to be able to bring your knowledges together to come to the best conclusions, which is really, really cool. So we got the they chew on a cotton ball, they rub this toothbrush like with thing on their gums. You're collecting tubes of blood. You're collecting vaginal samples. You're collecting rectal samples, and you're freezing it. So you're not really doing anything yet. You're freezing it because when you're done with all your patients, then you're gonna analyze and compare and contrast. That's what's coming next, right?
Right.
I'm with you.
And I want to also say one big motivation that we have towards all of our participants who participate in MAESTRO study, we want to give back the data, whatever that is available as a validated reports to benefit our participants. And sometimes with a small academic collaboration, there's just no way for us to like generate a patient or participant level of individual report because this is a more like a research project. It's literally for research only. But some proteomic data, or other like metabolomic kind of analysis that we're going to do this, we're going to run those testing through the rigorous companies that we're working with. And those are all CLIA certified company facilities, meaning that it's certified technique that they run those testing, and they are able to generate individual case report if you request to. We want to really find a way that can help our patient community by providing back those results, even though this is still a research project and there's a really fine line that we cannot really explain what it really means.
Yeah, you're giving as much as you can to help them with the
yeah,
results of these tests that you're able to
yeah,
provide to them that can help them in their experience.
yes.
Yeah, I mean, so so we talked about the 4 hour visit, and now this is essentially has been the past couple of years has been you interviewing and getting onboarded 200 patients, getting them on site, bringing them through this rigorous process, which is about to conclude in the next couple of months. Then you're going to go head down and start to do the deep dive research and investigation on all the samples and all of your surveys and all of the things you just discussed.
Yes.
So, one of the things that I can't help but think about is there are other entities out there who do things like this. So there's the Lyme Biobank out in California, who we've had on the podcast. There's LymeDisease.org, who has the My Lyme Data registry, where Lyme patients go on and fill out these surveys. And that's where we learned that fatigue is the #1 symptom for Lyme disease. And this is all, backed by these reputable groups. Have you guys ever thought about collaborating with others to scale and have a bigger reach for people so you can have more data points with the experts at MIT? Because LymeDisease.org is a great organization, but they're not researchers. They're not doctors. They're not you guys, right? But their information could certainly help you. So as an outsider looking in, I see this being an amazing facility, but I also see potential for improvement by collaborating potentially with others to feed the data engine that you guys are analyzing. What are your thoughts on that?
Absolutely. I'm 100% with you on that. This is where it's critical to collaborate. Research has to be a one project with a combined mindset of really help out this community. And this is when I get really thrilled. In fact, personally, whenever I attend some conferences, even the New York Fashion Show, to support the Lyme Foundation, I just find this so interesting to meet all these collaborators, and we get to share what we do. And that's the instant moment. Like, we have this many sample, we have this many data from the survey. Would you like to collaborate? And this is where, sometimes my social skills really shines on too. I love getting connected with all the collaborators. I do have a tremendous also respect to some of the researchers who are in the same field, and some of the researchers, it seems like we're doing very similar research, but actually there are things that we can collaborate together to compare our samples and continue to collaborate by discussing what we've discovered and how we can navigate this direction further. That's one thing that I'm always interested in by talking to other researchers in this particular area.
Yeah, I just want to share with you because I just love what you're saying. Yeah. So I attended, again, I work in the IT space, and I attended a cybersecurity conference by the New York Association in New York City. And this wonderful speaker who worked for the CIA and all these agencies was talking to us about how the federal government from the early 2000s forward interacts with organizations that are the most vulnerable and susceptible to cyber attacks. And they
Mm-hmm.
said banks are such prime targets where other nations and nation states are constantly targeting our banks to cripple our nation. And
Yeah.
this happens every day, all day long. And you don't really think about it. And the
Yeah.
relationship here is pretty cool because what they did is they had all these banks, let's say I'm making up a number, the 7 leading banks in America. They'd
Mm-hmm.
have quarterly calls with point people and they collaborate and share. Hey, this is happening from North Korea. This is happening from China. This is happening from Russia. What are you seeing? What are you seeing? This is what I'm learning. How are you? And even though they're competitors, they were forced to work together because they had to protect their interests without being crippled from a cyber attack. And it
Mm-hmm.
makes me think all of the researchers, the Monig Embers, the Mikki Tals, the Yuri Kims, do you guys collaborate? And if not, do you think it's a good idea because you're all so brilliant and you can share information and help each other, which ultimately helps our community. So what are your thoughts on that?
No, I love that idea. I would love to. And if there's any platform that we can securely even like exchange our data in the future, that would be a game changer. I'm so glad to hear that you're involved in this like IT field here because that's always my problem that I'm facing. And I always realize I would love to collaborate, but there's such a huge gap to share the information. That we've collected online, because of the institution does not have a completely different system. Just like a long COVID research project nationally, there's a recover study, the project going on where multiple institutions kind of gather to one website to share their biological sample profile or survey information together. Why don't we create some type of platform like this
Yeah.
to combine the information, but also for patients to also see based on their symptoms or what they've experienced what kind of, like what is most likely they should check, and based on their symptoms, what is it, where do they most likely belong to in terms of a large sample cohort that the information that we have in the platform. I think that would be also really helpful. I I really want to say one thing here. My job is always listen to my patient, and our 4-hour study visit met. 6 months later, I follow up with our participants. 1 year later, after the baseline visit, I still follow with our participant via Zoom meeting. Some of them are willing to come back to the longitudinal visit and to capture exact same testing data. It's been interesting to see the differences if one were to say, I tried this medication and really helped me significantly to lift out my brain fog. I'm able to actually function my job now. Or some people say, I have had a mold exposure recently and really I have a severe flare up. I'm in the midst of a severe brain fog or neurocognitive dysfunction. And depending on their report, what has helped them and what has flared their symptoms or triggered their symptoms, why don't we gather all this information and share to really learn about this particular, Lyme disease related chronic illnesses? I think that's where the critical collaboration is really pending here.
Yeah, and I think you're right, though. You need a secure platform, you need a hub that
Yeah.
you all feel comfortable sharing in. And then now, more than ever, with artificial intelligence and machine learning, you can leverage computer tools to be able to analyze it and provide you with feedback with the human involvement, the researchers, all the niche expertise all of you guys have in the research, world with all these different colleges and universities doing this work. If you had AI, machine learning, and all of your brains together in one platform, I think we'd be much further ahead than we are today. It's just how do we get there, right? And how do we
How do
accomplish
you get
that?
I mean, but one of my personal interests is how does the brain fog looks different in terms of the traditional traumatic brain injury group, How does it
Yeah.
look different when if it's associated with the infectious disease? So that's another collaboration that we can do. And also, I have a personal advocated feeling towards a pediatric population as I'm talking with a lot of my participants who recall their symptoms started when they're teenagers or they're talking about being 9 year old and they saw the tick bite, but they thought it was not a big deal. Didn't even tell their parents, And this
Yeah.
when the symptoms started. I want to really help out those pediatric population where their illnesses need to be adequately and properly addressed. And this kind of collaboration is actually being done. Pans and Pandas community. I meet with some of the experts there, and this is how we can also really address the root cause. Lyme disease can be one of the root cause as an infectious disease organism. So this is really interesting. Yes.
Yeah, and I'm just, it's so funny you're saying this because you're right. There's so many people. I mean, we're having Dr. Summer Del Signorion, and she's a pediatric Lyme specialist.
Mm.
What can she contribute to MIT and to, Yuri Kim and to Mikki Tal? How can you guys, if you were all to work together, how can that information help you, and how can your information help her? So I think that to me segues into my next question is what's next for you and what's next for Tal Research Center? We touched on additional collaborations, additional maybe tech, infusion into what you guys are doing. But, once you close, you're going to do your compare and contrast, and you're going to do your deep dive research. But what after that? You talked about a potential follow up or an additional research study down the road. So where are your heads at with the future?
Well, with the future, very near future, we are actually launching another study very soon. Once Maestro study closes, of course, with the analysis, we're going to navigate further direction where we can truly benefit our patient community here. One project we're really building on right now is based on what we find in the Maestro study. We want to really specialize looking into the brain fog symptoms in female participants in particular, and really trying to see the association with the hormone or menstruation cycle and compare this type of brain fog with infectious disease related brain fog versus just typical menopause brain fog, in the control group, or premenopause brain fog. So we just want to really we can always think about based on what we are doing already. And then the next kind of vision that we have is, like I said, Matt, I mean, like you mentioned, the collaboration is the critical key here. I would love to have opportunity to continue to collaborate with Dr. Monica Ember. There's also Dr. Sue Farber in Canada, who also specializes pediatric Lyme disease, or the fetus maternal transmission of the Lyme disease. So I would love to find the ways to really look into those particular things. And also with the PANS and PANDAS, the way I look at it is that, as I mentioned, I describe about eye tracking testing and EEG testing. And I always tell to the clinicians and researchers whenever I meet them in the research conference setting is that these are the non-invasive, and actually the eye tracking testing can be quite fun to some of our, pediatric population, non-invasive, but there are ways to perhaps measure these pediatric symptoms of the neurological or the conditions and be able to track over time without causing it so much of an invasion or pain. So I do have a strong advocacy that there are ways to measure these things, and I would love to continue to seek for a conversation with the more experts in the future.
Yeah, I just love how what you're doing in the clinician world with real world Lyme patients, seeing them in person, touching base with them, staying in touch with them, and then constantly funneling it back with Dr. Tal on the research side. I mean, you're bridging the gap between researchers, clinicians, and then everyday practitioners that are treating Lyme patients. And it just shows that patient voices are really shaping the future. So the patients are being heard. It shows, look, I think Maestro is a turning point for the Lyme community because it's giving us something that we've never had before. But for the first time, patients are being asked to they're not being asked to fit the data model, right? It's, hey, this is what science says. You have to fit this, otherwise we can't help you. The data is going to be built around the patients, and that's what you're doing there. You're building the data around the patients to find things that we currently don't know, and that's what's very much needed in our space. So, I do want to ask if you could just give us, if I had to say to you, what is the number one thing that gives you hope for the future of our Lyme community? What would it be?
Oh my goodness, that is a very hard question. Number one, hope. Did you say hope, right?
Yes, to give you hope for the future of our community, above and beyond what you said. I mean, you gave us a ton, but the number one thing, if you ran into somebody in a grocery store and it was the mother of somebody suffering greatly with Lyme disease, and she was telling you how much her child is suffering, what would you say to offer her words of encouragement and provide her hope for the future of her child?
I often say this, Beth, and I do say this from my absolute sincere bottom of my heart. If I can ever deliver that feeling to my participant whom I met for the first time via Zoom during consenting, I just say, please don't give up. I just say, please don't give up. Please do not give up. I know the frustration. I know that the I hear your voice and we're all in this together. I don't want our patients to give up because there are researchers like our team and other institution whom we meet always on a regular basis, and we really talk about how we can benefit our patients together. That's my voice. If I can speak up and if I can tell my patients or anyone even random person whom I face in the grocery store with their child being sick, I want to say, please don't give up. There will be answers.
You have given me hope and everybody listening to this podcast hope. The Lyme community is in a place that I've never seen it be in since 2014. We are, constantly making progress. We're not where we need to be, but we're in a pretty good place compared to where we were. And I'm thankful for that. And that is directly because of people like you, Yuri Kim, and Dr. Tal, and everybody else. So I am so grateful for you personally and everything you're doing. I can't thank you enough from the bottom of my heart. And from everybody at the Tick Bootcamp community, we just want to thank you, thank you, thank you. And thank you for joining our Tick Bootcamp podcast.
Thank you so much, Matt. It was wonderful. I also want to say, to not give up. And also, I'm absolutely grateful for all of our Maestro participants who contribute their precious time, their life to be part of our study. Their contribution in participation will do everything. And it'll be a game changer that once we close our study and be able to produce the data that can benefit the community and with throughout the publication, we really want to change the conception and social stigma surrounded with the chronic Lyme disease. And we hope to continue to do our work and bring more hopes and more exciting research to our patient community. I mean it.